All right, guys, this is video 802. We're going to talk about DNA replication. So we have talked about the fact that DNA sits in the nucleus, but we haven't talked yet about how it, first of all, copies itself for meiosis, and then how it forms proteins. And so what we're going to do today is we're talking about the process of replication. And what we mean by this is in mitosis, we talked about the cell life cycle. We said there's interphase, and then we go into mitosis where we have prophase, metaphase, anaphase, telophase. So if you remember, we had interphase has sections. So let's go back over that. Interphase had 3 divisions. So do you remember? All right, did you say G1, S phase, and G2? If you did, that's correct. So if you remember, it's a G, we grow, and then in S phase, we duplicate the genetic information. It was the S stood for synthesis, and then we grow some more, and then we go into mitosis. So this replication is what's happening during the S phase, which stood for synthesis. And so what we're doing is we're taking DNA and we're doubling our DNA. So we're going to double the DNA. And we have a diagram here that shows the steps. So we start off, we've got the parent molecule has 2 complementary strands of DNA. Each base is paired by hydrogen bonding with its specific partner, A with T and G with C. This is what we reviewed in the last video. I wanted to make sure you guys were good. So we have hydrogen bonds here in the middle. And if you remember, we talked about the fact that here we have covalent bonds. And covalent bonds are much stronger. They're much tougher than hydrogen bonds. Hydrogen bonds are relatively weak. So that's why the first step in replication is separation of the 2 DNA strands. When these guys, when the time comes, there's an enzyme that does this. It comes in and it causes these bonds to break. And again, that happens by an enzyme. You don't need to know that. It's a little above our pay grade. But when we separate the 2 strands, we call this unzipping the DNA. So the DNA gets unzipped. Now, this is, of course, you remember, where's the DNA found? Where does it live? Did you say nucleus? So this is taking place in the nucleus. This takes place in the nucleus. So in the nucleus, we've got our DNA. The enzyme comes along and unzips it. Now, floating around freely in the DNA or in the nucleus are free-floating nucleotides. So these are free-floating nucleotides in light blue. It looks green when I write on my blue on the yellow. So all of these are the free-floating nucleotides. So what you can see is that we have a strand of the original DNA. So in dark blue, we have the original. Here's the original. Here I have the new. So in light blue, I have the new. And what happens is as this strand of DNA gets unzipped, another enzyme comes along and helps to insert these new nucleotides. And so what you get is for each of the 2 strands, 50% of it is original and 50% is new. And there's a famous experiment that they did to show that this is how DNA replicates. What they did was they put a strand of DNA in the presence of a vat of a container that had they attached a radioactive tracer to the nucleotides. So they put a radioactive end on these free-floating nucleotides. so that they could tell, they could use instruments to detect how much radiation there was in each of the new strands of DNA. And what they found was, when they tested it, that 50% of the material was radioactive and 50% was non-radioactive. So they determined that this is how it happened. And you might say, well, how else could it have happened? I don't know, maybe it only copied one strand and then it broke away and this became a dormant strand, or, there were other theories as to how it could happen. But again, they were able to prove that through that famous experiment. I can't recall the name of it right off the top of my head. But what are our takeaways here? We're talking about the synthesis that takes place in interphase during the S phase. This needs to happen, takes place in the nucleus, and this needs to happen before mitosis. So this needs to take place before mitosis. Because if you don't double your genetic information, then your new cells aren't going to have the genes that they need and the DNA that they need. We take this weak bond, hydrogen bond in the middle. It gets unzipped by an enzyme. Another enzyme comes along and starts attaching in these free-floating nucleotides. And we end up with two exact strands. So if you look, we started with ATCGTAAT, and each of these is exactly the same, but they're 50% old, 50% new. All right, that was it's kind of a quick hitting video. There's not a lot of detail here, but yeah, now let's move on to the protein synthesis. All right, that's all for now.